Hey guys, so today we're going to talk about Mendelian inheritance. And so we've kind of already covered a lot of this in class. I'm kind of going a little bit out of order. But what I want you to take from this video is the fact that especially traits that are involved with humans, especially have more than one gene that will code for it. So you may not see the things you expect. So for example, I have brown hair, Barry has blonde hair, and our son Jace has brown hair, which we'd expect, and our son Jackson has blonde hair. So there's a little bit more that goes into this whole factor than just dominant and recessive. And I'm going to talk about all that stuff today. Remember that humans are a lot more difficult, and when we start getting into the human aspect, we'll see a lot more diversity present. So at the end of this video, you should be able to define probability, explain how probability is related to inheritance, describe how to use a Punnett square, explain how Mendel interpreted the results of his experiments, and finally describe complex patterns of inheritance. Okay, so before we can even get into the huge realm of Punnett squares, we need to talk about an idea known as probability. And you've probably heard of this term. I don't think it's a new term, but probability is the likelihood or chance that a certain event will occur. So you can think of a coin toss. If you have a quarter, you have a heads and a tails. There's a 50% chance that you'll get heads. Now let's say you get heads 10 times. And while you're thinking, well, there's no way I could get heads 10 times. The chances of getting heads 10 times is 50%. It's always going to be the same probability. Even if you say it's so unlikely, there's still always the same probability. So, but eventually, so I just gave you the example of flipping the coin 10 times, and the probability of getting the same side is 50%. But eventually, if you were to flip this coin like 1,000 times, you would probably get a lot closer to 50-50 results. So let's say you were to flip it 1,000 times, you might get something like 510 heads versus 490 tails. A lot more closer to 50% than when you flipped it 10 times and 100% of the time it was heads. So if we take those rules of probability or chance and we apply them to inheritance, we see the same kind of events occur. So remember when a zygote is formed, it's with the union of gametes and that's fertilization. And then to create those actual gametes is done during meiosis. So I'm going to use an example when we're talking about gametes and the formation. Remember, this is done during meiosis. So gametes, there's one from your mom, the egg, one from your dad, the sperm. And so let's assume that your mom is heterozygous for a certain allele. So that genotype would be capital, lowercase. During meiosis, the homologous chromosomes and the alleles that are carried segregate and go to different gametes. That means that capital and that lowercase are separating from one another. So one cell will get the capital, the other cell will get the lowercase. So about half of the gametes produced will have that capital B, and half of the gametes produced will have the lowercase b. So 50% chance that you will get one of those alleles from your mom. So like I said, same exact way that we do coin tosses is how we can look at homologous chromosomes. Now remember, your mom has 23 chromosomes that she has to pass on to you. So you don't know what you're getting from your mom or dad. And then there's another 50% chance when it comes to probability and fertilization. So the gametes that were formed, remember, we split the alleles. So half are capital B, half are lowercase b. Now there's another 50% chance for which of those gametes is actually going to be the one that actually undergoes fertilization. So again, all of these chances are just due to chance. Like it's just a random event. And I guess that's what makes this whole process really spontaneous, and what makes you so special is the fact that you are incredibly unique. You were meant to be who you are just based off of chance and time and every single component that came up in life at that particular moment. So again, all of genetics is basically based off of probability, and the chances that you are who you are and the traits that you have are because of a flip of a coin, basically. And so geneticists after some time, geneticists were studying a lot of traits, and they wanted to find a way that could help them determine traits of future offspring. And so a Punnett square is a very nice way to do that. A Punnett square is a chart that allows you to easily determine the expected percents of different genotypes in the offspring of two parents. And we've done Punnett squares in class already. We've done very basic Punnett squares. And so when we crossed a homozygous dominant with a homozygous recessive, we saw that 100% of the plants exhibited the dominant trait. Then when we crossed those heterozygotes, we saw a completely different percentage value. So Punnett squares are a great way to determine what you're expecting to see. And so Mendel, he didn't always get exactly 75% and 25% for his results, because remember, he's studying thousands and thousands of pea plants, but he probably got something along the lines of 76 and 24%. Or even like 80% and 30%, still really close to one another and really close to those percentages of expectation. So again, Punnett squares are very good at predicting offspring. They are also really good at determining missing genotype. So let's say you have a parent plant with purple flowers and a parent plant with white flowers. Because the lowercase b, or the white, is recessive, you know that white flower always has to have the lowercase genotype. But you don't know if the parent is either heterozygous or homozygous dominant. So what you can do is you can create a Punnett square to figure out what that parent is. But you would also need the results of the offspring, so you can't just say or take a guess, oh, it is heterozygous because I think it's heterozygous. So once the offspring have formed, then you could fully predict what the parent's genotype is. So like I said, we've crossed a recessive white with a purple parent, and we don't know if that's heterozygous or homozygous. So if the offspring come out and they're all purple, then you know that the parent was homozygous dominant. If the offspring come out and 50% are purple, 50% are white, then you know that the parent is heterozygous for that trait. And I'm going to show you what I mean by this in class. So don't worry if you're not visualizing what I'm saying, but hopefully this is making more sense with all these words I'm throwing at you. And I didn't include this on the slide, but I am going to mention that you can make Punnett squares for more than one characteristic. We're going to be practicing first with two characteristics. There are a couple things I want you to write down. So when you have two characteristics, you're going to have two letters. So I'll write that, and then I'll show you the rest of this in class. So you'll have two letters for two characteristics to represent each trait. My handwriting sucks, sorry. I can't believe I forgot this. So two letters to represent each trait. So for example, if we're looking at pea plants again, and we want to look at, green pod color and yellow pod color. So those two, that's the pod color is the trait. So green is one color and yellow is the other. And we'll represent that by a capital G. And then F represents pod form, which would be either like a full pod form or like a constricted, crunched up pod form. And so I know that doesn't, maybe you can't visualize that, but those are two different traits. So we would have to use two different letters to represent that. G being for the pod color, F being for the pod form. And I'm not going to do that on here. I just don't want you to feel overwhelmed. We are going to practice and practice and practice. And by the time we're done, you'll be able to do as many Punnett squares as you feel you need to do. So let's go ahead and talk about non-Mendelian inheritance. And so the first thing I want to mention is that Mendel was super lucky. He didn't know anything about chromosomes, but what he did do was he happened to pick traits that were not on homologous chromosomes, meaning traits that were not inherited with one another. So, for example, when he picked color versus pod form, those were non-homologous traits. So he could very well see the two traits independently of one another. So if Mendel didn't actually do this and he wasn't that lucky, his law of second law of inheritance, the law of segregation, would not actually be a thing. So, or law of independent assortment, excuse me, would not actually be a thing. So it's pretty lucky, I would say. And he is the father of genetics, so he had something going on for him, but he was also very, very lucky. So there are different types of dominance, or excuse me, not dominance, but inheritance. I also want to talk about concepts known as codominance and incomplete dominance. And so what those are is that a trait may be controlled by one gene with two alleles, but the two alleles may have a different relationship than simply dominant and recessive. And those types of relationships are known as codominance and incomplete dominance. So again, there's still two alleles present, but there's not a dominant recessive relationship. They have a codominance, and if you are codominant, that means that they share, or the traits are expressed equally in the phenotype of the heterozygote. So for example, if I have two parents, a red and a white flower color parents, and then they pass on, and then the offspring become red and white, that's codominance. They're sharing those traits. So an equal share of those traits. In comparison to incomplete dominance, where there's not true dominance at all, you have more of a mix. So for example, if you have two parents, let's say we have a pink offspring and the parents were red and white. So that offspring is obviously more of a mix than either side is more dominant or the other. To continue on with non-Mendelian genetics, we have what's known as multiple alleles. So where there's more than two alleles that express for something, great examples of this would be blood type. So for example, ABO blood type in humans, so we have A, B, and O. There are three common alleles for the gene that control this characteristic. So for example, I'm O positive and my husband's A positive. If, for example, if I was B and my husband was A, the chances of our kids inheriting both of those traits is pretty likely. So the allele for type A is codominant with the allele for type B, meaning neither one is more dominant than the other. And both alleles are dominant to the allele for type O. So I'm a type O blood type, my husband's type A blood type, both of our children are A blood type. So the A blood type is dominant over the O, B blood type dominant over the O as well. So the possible phenotypes that you could have for those three different alleles is A, B, AB, and O. And we're going to do that. I would think that's more of an advanced issue, a problem. So as we progress into this, you'll see what I'm talking about. Polygenetic characteristics means that these characteristics are controlled by more than one gene, and each gene may have two or more alleles. So the genes may be on the same chromosome or on non-homologous chromosomes. Great examples of these would be like height and skin color are very much polygenetic characteristics. So the chances of you inheriting the same genes on homologous chromosomes because they're just close to one another is pretty likely. But remember, we have crossing over. We have non-homologous genes that are a factor as well. So that is why you look unique in comparison to anybody else in this entire world. Another thing to consider is effects of the environment on phenotype. So if you were to be born into an environment that wasn't conducive to achieving your 100%. So what I mean by that is, let's say you're in a third world nation and you have two very tall parents, but you just don't grow very tall. The chances of your nutrition affecting you or not being able to get modern medicine can affect your genetics. So you may not fully grow as tall as you should just based off the fact that there are environmental factors that are affecting you. Food availability, lack of modern medicine. Another one, like I said before, skin color is another determinant of the environment. So exposure to ultraviolet radiation can increase the amount of pigment in your skin and make you appear darker. And you know this, you go out in the sun and you either tan or you get really, really red. So that is all I have for you in this video. Hopefully you're starting to enjoy this more. I know that the last chapters you haven't really enjoyed. If you have any questions, please ask me on Edpuzzle. Thanks.